Good evening, everyone. So, myself Sonam Pahuja. Today we are here for the live session week 7, and I'll be taking the course for NPTEL Cell Biology, Cellular Organization, Division, and Cellular Processes. So, we will cover the syllabus of week 7, that is cell death, aging, senescence, apoptosis, and the cancer cell. So, starting from the question number 1, given that CED9 protein can bind to and inactivate CED4, which of the following would cause apoptosis? The CED9 appears to be binary switch that modulates cellular survival and apoptosis in nematodes. Which of the following options is correct? If anyone wants to answer the question, please speak up, as I won't be watching your chat box. So please speak up if you know the answer.
Option 1.
Sorry for the Sorry for the inconvenience. So if you people know the answer, please speak up, okay? Is the screen visible? So the question was given that CD9 protein can bind to and inactivate CD4. Which of the following would cause apoptosis? The CD9 appears to be a binary switch that modulates cellular survival and apoptosis in nematodes. So which of the following options would cause apoptosis? Anyone?
Option 1.
Option 1. It is option 2. How? Can you explain why were you saying option 1? Please, can you speak up?
Mm, Um, ma'am, yes, sorry, CD9 gene was introduced by CD3 and 4, and then it will lead to, I think, activation of apoptosis. It was activation of apoptosis or inhibition.
What is the function of CD9 gene? CD9 gene or protein, what is its function? It inhibits apoptosis in nematodes. Okay. It inhibits apoptosis in nematodes. So they are saying which of the following would cause apoptosis? The CD9. Here they are talking about CD9 mutant. It appears to be a binary switch that modulates cellular survival and apoptosis in nematodes. So if we are saying that loss of function mutants of CD9 gene, if CD9 causes inhibits apoptosis, so CD9 mutant have inappropriate apoptosis that would normally survive. Okay. So loss of function mutant will have inappropriate apoptosis and they will survive, while gain of function mutant of CD9 are defective for apoptosis. As CD9 inhibits apoptosis, so they will die. Okay, so here it is saying this appears to be a binary switch that modulates cellular survival and apoptosis in nematodes. Okay, so here loss of function of CD9 gene. If it cause apoptosis, loss of function of CD9 will cause apoptosis and not the gain of function. Okay, clear to all? Is it clear?
Ma'am, why not option number 4 then? Point mutation of CD4 gene.
What will point mutation cause? Is it necessary that point mutation causes the complete loss of function?
No, ma'am. It might lead
It
to.
might, right? So we don't know what kind of point mutation it is. So to be very sure, so loss of function will be the correct option here. Okay. CD9 क्या करता है? एपोप्टोसिस को इनहिबिट करता है. ओके, तो CD9 नहीं होगा तो एपोप्टोसिस होगा. So it is saying which of the following would cause apoptosis. So loss of function of the CD9 gene. Okay, clear?
Yes, ma'am.
Okay. So to be clear about this concept, here we are talking about Cenorhabditis elegans, that is C. elegans. It is a nematode discovered three crucial elements of the cell death system. Cells are killed by CD3, 4, 3 और 4 से cells kill होंगे, whereas CD9 जो है it will prevent the cell death, as it is saying that it prevents apoptosis or cell death. It was discovered that CD9 and BCL-XL, which is a cell death regulator from the BCL2 family, interact with each other and block CD4 activity. Additionally, research showed that CED4 and its mammalian counterparts, flies and eyes, as well as CD3, can interact at the same time. So, by biochemically connecting CD9 and BCL2 family to CD3 and ICE family of pro-apoptotic cysteine proteases, CD4 thus plays a crucial role in the cell death cascade. So, talking about programmed cell death, it is a major aspect of metazoan development. During the development of the nematode C. elegans, 131 of the 1090 cells generated undergo PCD, programmed cell death. Mutations affecting the process of programmed cell death in C. elegans have been isolated and have defined 14 genes. So here loss of function, mutations in the gene CD3 and 4, and gain of function in the gene CD9 or overexpression of the wild type CD9 can prevent programmed cell death. So it was saying that which of the following would cause apoptosis? So loss of function of CD9, or it will be gain of function of CD3 and 4. Okay, as it is saying that it can prevent programmed cell death, so loss of function of CD3 and 4 and gain of function of CD9, it can prevent programmed cell death, and vice versa. If it is saying that which promotes programmed cell death or which promotes apoptosis or causes apoptosis, so it will be gain of function of CD3 and 4 and loss of function of CD9. Now clear to all this?
Now, what is wild type CD9 gene?
Wild type C9, CD9, that whole protein is functional, that it is functioning properly like wild type. Wild type is normal control protein, no kind of mutation. Here loss of function or gain of function or point mutation, it is different variants of mutations. So in wild type CD9 means there is no kind of mutation, it is control. It is control CD9 protein without any mutation. Okay.
Means mutation can't occur in that gene, or it can occur, but it's not occurring.
You are talking about what?
This sentence, it's written that in the gene CD9 or overexpression of the wild type CD9 gene can prevent programmed cell death.
Yes. So it is saying overexpression of the wild type CED9 gene. Wild type CED9 gene means normal. It could be overexpression of CED9 gene. It means no other variant. It could be overexpression of the mutated, any like mutated, like if any mutation of CD9 gene that is already known, that any constitutively active variant of CD9 or any dead variant of CD9, like any point mutation of amino acid causes uh, like programmed cell death. It is saying that suppose lysine to asparagine of CD9 point mutation can cause the programmed cell death, and if we overexpress that point mutation of CD9 gene, it can prevent cell death, right? So it is saying overexpression of the wild type CD9 gene, no mutation, normal CD9 gene. Okay, so if it is loss of function of CD9 gene, It can cause apoptosis, and if it is gain of function or overexpression, it can prevent PCD. It is normal wild type CED gene. Normal wild type means no mutation. Okay.
Okay.
Coming to next, which of the following factors is known to be involved in postponing PCD in serial alluron until endosperm mobilization is complete? Which of the following factors is known to be involved in postponing PCD in serial alluron until endosperm mobilization is complete? Anyone? Anyone knows the answer? Is it gibberellic acid, abscisic acid, acidic pH of the vacuole, CGMP mediated signaling transduction pathway? Screen is visible, right? Anyone knows this answer? No one? Okay. So it is abscisic acid. So let me explain. Abscisic acid is the factor that postpones the programmed cell death in serial alluron layer until endosperm mobilization is complete. As we all know that ABA is a hormone that slows down the process of alluron cell death. Alluron cells are viable in mature grain but undergo programmed cell death when germination is triggered. So, cell death in barley alluron occurs when cells become highly vacuolated and lose plasma membrane integrity. So, when we add ABA to cells pre-treated with GA, it can delay the cell death. And also, ABA induced protein can inhibit the GA mediated programmed cell death in serial alluron layers cells. So, programmed cell death is also known as apoptosis, and this ABA slows down this process. Okay, so it is saying which of the following factors is known to be involved in postponing PCD in serial alluron until endosperm mobilization is complete. So it is abscisic acid. Coming to next, PCD plays an important role in development of barley alluron. The following statements are made with respect to involvement of various phytohormones and signaling molecules. So which of the following combinations of statement is correct? Anyone?
Option 3.
Option 3, A and B. Correct. I recently told in the previous question that ABA postpones PCD and GA promotes PCD. And no other things these are involved like cyclic GMP or nitric oxide. So,
Ma'am, these topics were not there in that lectures, means
what?
like these topics like hormonal effect on programmed cell, that these topics were not in that lectures in NPTEL.
Yes, yes. It's extra as it covers apoptosis and cell death. So, I have involved these topics as well.
Okay.
Okay.
Hmm.
So, it is in your syllabus, although they have not covered it in the assignment. So, I have taken from other sites. Okay. So, alluron cell death does not follow the apoptotic pathway found in the animal cells. So, hallmark of apoptosis including internucleosomal DNA cleavage, plasma membrane, and nuclear blebbing. Formation of apoptotic bodies are not absorbed in dying alluron cells. PCD in barley alluron cells is accompanied by the accumulation of spectrum of nuclease and protease activities and loss of organelles as a result of cellular autolysis. So these were the statements where it is saying GA promotes PCD. As we know that serial alluron cells are specialized endosperm cells that function to synthesize and secrete hydrolytic enzymes that break down reserves in the starchy endosperm. So, unlike the cells of the starchy endosperm, alluron cells are viable in mature grain but undergo PCD when germination is triggered or when isolated alluron layers or protoplasts are incubated in GA. So, this treatment is true that it promotes the PCD. And how ABA postpones PCD? It slows down the process of alluron cell death, and isolated alluron protoplast can be kept alive in media containing ABA for up to 6 months. So, cell death in barley alluron occurs only after cells become highly vacuolated and is manifested in an abrupt loss of plasma membrane integrity. So, this statement is also true. So, cyclic GMP signaling postpones PCD. It is not correct. And nitric oxide scavenger delays PCD. It is a We know that nitric oxide is a freely diffusible gaseous free radical and an important signaling molecules in plants and animals. So in plants, NO influences aspects of growth and development and can affect plant responses to stress. So nitric oxide acts as an antioxidant and it delays PCD in barley alluron layers. Okay, so NO it delays PCD. NO
Ma'am,
scavenger.
so
NO delays PCD. So it is saying NO scavenger. Okay. If it is NO, it could be correct. If it NO scavenger, it promotes PCD. Okay. As NO acts as itself acts as an antioxidant, itself acting as a scavenger. So NO scavenger promotes PCD. But NO delays PCD. So this statement is also not correct. So the correct statements are this A and B.
ma'am,
Yes.
can you move to that slide, the previous?
Yes.
Here, this uh, this in the first paragraph,
Yes.
this PCD in barley alluron cells is accompanied, means it results in the spectrum of nuclease and protease activities rather than DNA cleavage, plasma membrane, which are the hallmarks of apoptosis.
Yes.
The here the features are different regarding apoptosis, or this means apoptosis in Aileron cells will lead to a spectrum of nucleus and protease activity. They will not lead to plasma membrane like DNA cleavage or plasma membrane and nuclear blebbing.
It is kind of same. Like obviously nuclease and protease activity will be there. If nuclease and protease will be present, there is accumulation of these. It can cause the protein degradation, protease, and also nuclease will cause nucleic acid degradation. So it is kind of same thing. And loss of organelles, we know that as a result of cellular autolysis. So it is no different than that. Although plant and animal systems are different, nuclear blabbing and whatever you were saying, it is different. But the function is same, like degradation of organelles, loss of organelles, and degradation of these nucleic acid and proteins will occur. Okay. Any doubts?
And ma'am, after the abscisic acid is kept while experimenting till 6 months,
Yes.
then the cells will become vacuolated and cell death will occur
Yes.
in case of abscisic acid. But till 6 months, they will, they will inhibit the growth of, means they will inhibit cell death.
Yes, they inhibit.
But
That's
after
why
six
we are saying
months,
that it postpones PCD.
but after 6 months, it will completely leads to death of that cell. In the
Yes.
second line, it's written that cell death in barley alluron occurs only after cell becomes highly vacuolated and is manifested in an abrupt loss of plasma membrane integrity. Means
Yes,
after
yes.
6 months, the cells will become highly vacuolated and then it will lead to cell death.
It's not
But
like
if,
after 6 months. It is saying that up to 6 months, it like it can postpone the PCD. It can postpone the PCD up to 6 months. Okay.
but
So
after 6 months, it will lead to death of that cell.
every enzyme or every hormone, it has some like activity, right? Even
Hmm.
antibiotic or something, it also has some activity, like sometime it will degrade, right? So if it is capped in ABA, then up to 6 months it can postpone PCD. It has been validated, right? So 6 months is not a small time that it can delay, right? Okay. Should we move ahead?
Okay, ma'am.
Okay. So what is apoptosis? Simple question. Answer?
Option 3. Program cell death.
Correct. Which of the following is not a phase of apoptosis?
Amplification.
Sure?
Maybe, ma'am.
Why?
Just guessing because execution initiation to initiation means it will apoptosis will get started, and execution means finally it has to be executed. And in the case of clearance, At last, the phagocytosis will take place, means the cell debris which will be left or fragmented cells will be eaten up by phagocytosis, but amplification might not be the process.
Yes, correct. It is amplification, right? So we have different phases of cell cycle. It is like induction, early phase, mid phase, late phase, and also we have execution phase, we have initiation phase, and we have clearance phase. So first of all, which phase? Starting from which phase? Initiation, right?
Initiation.
Right. So what happens in initiation?
Cell will get prepared for apoptosis after certain antibody or any foreign substance or any TNF like tumor necrosis factors or more was just like that type of factors. If start that, then they will initiate the apoptosis by
You
bringing certain like caspases or CDs they will bring, and then
yes. You might have heard of apoptotic pathways, which is extrinsic and intrinsic, right? So they are initiated by the activation of death receptors, which are present at the cell surface, right? So initiation of apoptosis is tightly regulated by activation mechanisms, because once apoptosis has begun, it inevitably leads to the death of the cell. So initiation, it is a phase of apoptosis. Then coming to execution, it is the activation of execution caspases that begins this phase of apoptosis. Execution caspases में क्या होता है? They activate cytosolic endonucleases, which further degrades nuclear material, and proteases that degrade the nuclear and cytoskeletal proteins. Okay, तो एग्जीक्यूशन भी हो गया. अब क्लियरेंस. क्लियरेंस क्या होता है?
all the debris which will be generated after apoptosis will be cleared by uh, macrophages or phagosomes.
Yes. And it has no role with the amplification, right? Moving on to the next, which of the following is an example of an extrinsic apoptotic pathway?
DRE, no, I mean DRP, death receptor pathway.
Yes, correct. It is death receptor pathway. Can you explain this?
Ma'am, actually, in extrinsic pathway, death receptor pathway was there in the video, and in intrinsic pathway, I think it was. So, explain means the process.
Yes, you can explain extrinsic and intrinsic, and then come to the example.
Extrinsic and intrinsic. Mm. T. Can't explain.
Can't explain. Okay. So what is extrinsic pathway, anyone? Extrinsic means this pathway of apoptosis, it begins outside the cell when conditions in the extracellular environment determine that a cell must die. It means extrinsic means outside the cell. Okay, intrinsic में क्या है? It begins when an injury occurs within the cell and the resulting stress activates the apoptotic pathway. So in both these pathways of apoptosis, signaling results in the activation of a family of cysteine proteases, which are named as caspases. So these can act in a proteolytic cascade to dismantle and remove the dying cell. Okay, एक्सट्रिंसिक कहाँ हो रहा है? आउटसाइड द सेल, जहाँ पे सिग्नल बाहर से आ रहा है कि सेल मस्ट डाई. इंट्रिंसिक क्या है? इंजरी हुई एंड इट अकर्स विद इन द सेल और अंदर से सिग्नलिंग पाथवे एक्टिवेट हुआ. Okay, and it activates further proteases like caspases that act in a proteolytic cascade to dismantle and remove the dying cell. तो इसमें P53 पाथवे, माइटोकॉन्ड्रियल पाथवे और रिसेप्टर पाथवे. तो इंट्रिंसिक पाथवे की अगर हम बात करें तो अभी हमने देखा व्हेन एन इंजरी अकर्स विद इन द सेल, तो इंट्रिंसिक स्ट्रेसेस क्या होंगे? लाइक ऑन्कोजीन्स होंगे, डायरेक्ट डीएनए डैमेज हो रहा है, हाइपॉक्सिया हो गया, सर्वाइवल फैक्टर डेप्रिवेशन. these all can activate the intrinsic apoptotic pathway. तो P53 हमारा किससे रिलेटेड है?
Intrinsic.
Yes, right. P53 is a sensor of cellular stress and it is a critical activator of intrinsic pathway. Okay, तो P53 इंट्रिंसिक हो गया. माइटोकॉन्ड्रियल पाथवे
Intrinsic.
ये भी इंट्रिंसिक हो गया. क्यों हुआ ये इंट्रिंसिक? क्योंकि रॉस रिलेटेड हो गया. मतलब कुछ भी अगर हमारे ये P53 या कुछ भी एपोप्टोसिस रिलेटेड जो भी P53 के टारगेट्स हैं वो क्या करेंगे? रिएक्टिव ऑक्सीजन स्पीशीज प्रोड्यूस करेंगे. तो रॉस जब इंक्रीज होगा इट कैन लीड टू ऑक्सीडेटिव डैमेज टू ऑल माइटोकॉन्ड्रियल कंपोनेंट्स. तो माइटोकॉन्ड्रियल पाथवे जो है वो हमारा इंट्रिंसिक एपोप्टोटिक पाथवे में आएगा. क्लियर? नाउ एक्सट्रिंसिक पाथवे. As we know that it begins outside the cell when conditions in the extracellular environment determine that a cell must die. तो इसमें एक होगा रिसेप्टर मीडिएटेड जिसमें हमारा डेथ रिसेप्टर पाथवे आ जाएगा. एक आएगा TNF सुपर फैमिली पाथवे. सुना है?
Yes, ma'am.
और क्या आएगा? FAS पाथवे. सुना है?
Hmm.
और AKT पाथवे. सुना है? AKT पाथवे. These are all receptor mediated apoptotic pathways. Death receptor pathway, TNF, FAS और AKT pathway. और ये एक्सट्रिंसिक है. एक्सट्रिंसिक में एक हमारा मेम्ब्रेन रिसेप्टर मीडिएटेड होगा या फिर साइटोटॉक्सिक होगा, मतलब विद इन लाइक सेलुलर स्ट्रेस. और सेलुलर स्ट्रेस में आएगा हमारा अगेन माइटोकॉन्ड्रियल वाला भी, like mitochondrial membrane permeabilization, not mitochondrial pathway. It is MMP, mitochondrial membrane permeabilization. पर डेथ रिसेप्टर पाथवे की बात हो रही है, तो ये हमारा एक्सट्रिंसिक एपोप्टोटिक पाथवे का पार्ट है, वाइल अदर टू आर इंट्रिंसिक एपोप्टोटिक पाथवेज. कमिंग टू
तो
नेक्स्ट.
मतलब P53 के कारण ही माइटोकॉन्ड्रियल पाथवे परफॉर्म होता है.
यस यस.
Because it leads to ROS.
यस. Other proteins you might have heard of caspases, which are also released from the damaged mitochondria. Okay. So interaction of BCL family members जो सुने होंगे, और ये जो डैमेज माइटोकॉन्ड्रिया से केस्पेजेस निकलेंगे, ये आपस में इंटरेक्ट करते हैं, और ये इंट्रिंसिक एपोप्टोटिक पाथवे के पार्ट है. तो ये स्पेसिफिक माइटोकॉन्ड्रियल पाथवे होगा. ओके. एंड ऑल्सो जो P53 से रॉस रिलीज होगा वो भी. फाइनल, it is like damage to mitochondrial DNA disrupts the mitochondrial oxidative phosphorylation which can contribute to number of human diseases. So mitochondrial pathway अगर है तो वो हमारे इंट्रिंसिक का पार्ट है विद इन द सेल. Coming to next, which of the cellular processes is regulated by apoptosis?
Tissue repair.
Tissue repair. Which cellular processes is regulated by apoptosis? So apoptosis covers all these: migration, division, and tissue repair. Why? एपोप्टोसिस में क्या हो गया? PCD mechanism, and it plays a crucial role in regulating number of cells in a tissue by eliminating unwanted or damaged cells, effectively controlling cell division. सेल डिवीजन हो गया. सेल माइग्रेशन तो कंट्रोल करेगा ही कि उधर से उधर मूव, दूसरे जगह मूव करना है या नहीं करना है. एंड रिपेयर तो यू वेरी वेल नो कि apoptosis can be involved in tissue repair by eliminating damaged cells and facilitating the regeneration of new ones. So all these three options are correct. Okay. Cell migration, cell division, and tissue repair, all these processes are regulated by apoptosis. Which organelle plays a crucial role in initiating apoptosis?
Mitochondria.
Mitochondria. Sure?
Yes, ma'am.
Right, correct. So as we know that mitochondria are considered central to the intrinsic pathway, they release pro-apoptotic factors when a cell is signaled to undergo PCD. लाइक माइटोकॉन्ड्रिया की आउटर मेम्ब्रेन में जो परमियलाइजेशन फैक्टर्स होते हैं. सो आउटर मेम्ब्रेन ऑफ द माइटोकॉन्ड्रिया परमिएबल बन जाता है, रिलीजिंग प्रोटीन्स दैट ट्रिगर द एपोप्टोटिक कैस्केड. We will study in the end the whole pathway that BCL2 family proteins are also involved and caspase will get activated in this. So how this extrinsic and intrinsic pathways are followed, we will study it. Which enzyme is responsible for cleavage of DNA during apoptosis?
CAD.
CAD. Correct. DNA ligase क्या करता है?
दो स्ट्रैंड को जोड़ते हैं.
लाइगेशन करेगा. पॉलीमरेज क्या करेगा?
और तो स्टार्टिंग में हेल्प करता है, मतलब जितना भी न्यूक्लियोटाइड्स है वो सब लाने में.
पॉलीमरेज. What is the function of DNA polymerase?
डीएनए पॉलीमरेज किसी एक उसको लाता है और फिर वो हेल्प करता है जो जीएनटीपीस को लाने में, न्यूक्लियोटाइड्स को लाने में. सिग्मा फैक्टर.
DNA हेलिकेज.
हेलिक्स मतलब दो स्ट्रैंड को के बीच में जो न्यूक्लियोटाइड्स है उसके बीच में जो हाइड्रोजन बॉन्डिंग है वो फॉर्म करने में हेल्प करता है.
और CAD क्या करेगा?
दिखाई.
हाँ जी. क्लीवेज ऑफ DNA ड्यूरिंग एपोप्टोसिस. सो डबल स्ट्रैंड स्पेसिफिक एंडोन्यूक्लियस है ये. इट इज़ काइंड ऑफ रिस्ट्रिक्शन एंडोन्यूक्लियस. बाकी सब क्यों बोले लाइगेज, पॉलीमरेज, हेलिकेज? क्योंकि यहाँ पर हम CAD को एक रिस्ट्रिक्शन एंडोन्यूक्लियस की तरह ट्रीट कर रहे हैं. जो डबल स्ट्रैंड स्पेसिफिक एंडोन्यूक्लियस है that breaks the genomic DNA of dying cells. Okay, so CAD is activated by caspases, which are cell death proteins, and it cleaves the nucleosomal spacer regions, which leads to chromatin condensation. CAD is complexed with an inhibitor of CAD in non-apoptotic cells. Other functions क्या होंगे इसके? Cellular senescence में involved है. CAD activation is a feature of sublethal signaling in apoptotic pathway and also play a role in cellular differentiation and cancer. Okay. Coming to next, which of the following is a pro-apoptotic protein?
BCL2.
ये पूरा पढ़ लिया है ना? अच्छे से पढ़ के आए हो आज. गुड. इट इज़ BCL2. BCL2 का जो फैमिली है प्रो एपोप्टोटिक प्रोटीन की, it can initiate apoptosis by blocking the anti-apoptotic activity of BCL2 and BCLXL. ओके, तो BCLXL क्या है हमारा? क्या एंटी एपोप्टोटिक है इसमें और क्या प्रो एपोप्टोटिक है? जी, डिफरेंस क्या है प्रो एपोप्टोटिक और एंटी एपोप्टोटिक में?
Anti-apoptotic prevents cell death, but pro-apoptotic leads to cell death earlier only before apoptosis.
Right. So which of these factors are involved? P53 क्या करता है? Is it pro apoptotic or anti apoptotic? P53 से रिलेट करते हैं. P53 वेरी कॉमन, राइट? क्या है P53?
Pro-apoptotic.
करेक्ट. P53 प्रो एपोप्टोटिक है. कैस्पेस 3 क्या है?
Besides pro-apoptotic.
Caspase 3 is a frequently activated death protease. It is kind of protease, right? अब यहाँ पर BCL2. BCL2 एक फैमिली ऑफ प्रोटीन है. BCL2 क्या होता है? BCL2 होमोलॉजी और BH डोमेन जो हम कहते हैं. सुना इनका कुछ? आउटर माइटोकॉन्ड्रियल मेम्ब्रेन में इनका क्या रोल होता है, इन दोनों का? जो 3 मैंने मार्क किए हैं, दीज आर एंटी एपोप्टोटिक. नाउ लेट मी एक्सप्लेन हाउ. MCL1 और BCL2 जो होते हैं एंटी एपोप्टोटिक हैं, राइट? दे मॉड्यूलेट सेल वाइबिलिटी एट द आउटर माइटोकॉन्ड्रियल मेम्ब्रेन. ओके. अब BCL-XL क्या है? BCL2 और BCL-XL ये दोनों एंटी एपोप्टोटिक हैं, राइट? और प्रो एपोप्टोटिक प्रोटीन का क्या काम होता है? दे इनिशिएट एपोप्टोसिस बाय ब्लॉकिंग द एंटी एपोप्टोटिक एक्टिविटी ऑफ दीज BCL प्रोटीन्स. इज इट क्लियर? प्रो एपोप्टोटिक प्रोटीन जो है वो एपोप्टोसिस इनिशिएट करेगा एंटी एपोप्टोटिक जो एक्टिविटी होती है इनकी, उसको ब्लॉक करके. कैसे करेगा ब्लॉक? बाय बाइंडिंग टू देयर माइटोकॉन्ड्रियल बाइंडिंग साइट्स और बाय ट्रिगरिंग द एक्टिवेशन ऑफ प्रो एपोप्टोटिक बैक्स और बैक. सो इसको एक्टिवेट करके बाय ट्रिगरिंग द बाय बाइंडिंग टू माइटोकॉन्ड्रियल बाइंडिंग साइट or by triggering the activation of pro apoptotic BAX or BAC, BAK. तो ये दोनों हमारे प्रो एपोप्टोटिक हैं, और अदरवाइज BCL की ये जो फैमिली है ये एंटी एपोप्टोटिक है। तो प्रो एपोप्टोटिक एंटी एपोप्टोटिक की एक्टिविटी को ब्लॉक करके और फर्दर प्रो एपोप्टोटिक को ट्रिगर करके काम करता है और एपोप्टोसिस कॉज करता है।
बैक्स का काम क्या है?
बैक्स के पास एक प्रो एपोप्टोटिक एक्टिविटी है, प्रो एपोप्टोटिक प्रोटीन है वो। बैक और एक थर्ड टाइप ऑफ और होता है विच इज़ बिड, BID। ओके, इनके पास प्रो एपोप्टोटिक एक्टिविटी होती है।
ये लोग भी मेम्ब्रेन, आउटर मेम्ब्रेन नहीं प्रेजेंट है बैक्स।
जो BCL2 की फैमिली होती है प्रोटीन्स की, उसमें अलग अलग BH होते हैं, लाइक BH3 ओनली जो होते हैं प्रो एपोप्टोटिक प्रोटीन्स, उसमें अलग अलग मेंबर हैं। ओके, तो जो एंटी एपोप्टोटिक हैं वो तो हमारे आउटर मेम्ब्रेन पे हैं।
इसका कोई YouTube वीडियो या ऐसा कुछ?
Yes, I will show in the end. Okay, remind me once.
Okay.
Okay, so for the time being we will move ahead without wasting time. Okay, so which signaling pathway is commonly associated with apoptosis? बताया था ये अभी बीच में एक बार।
TGF बीटा पाथवे।
हाँ जी, TGF बीटा पाथवे।
फास्ट और फास्ट वाले पाथवे। TGF नहीं, ट्यूमर TNF होता है, TNF पाथवे।
इसका आंसर क्या होगा?
फास्ट एल पाथवे।
करेक्ट। अभी बताया था ना एक पीछे क्वेश्चन में, कौन से क्वेश्चन में बताया था? इस वाले में डिफरेंट पाथवेज जब बताए थे मैंने, रिमेंबर? और बाकी फंक्शंस क्या है, बाकी पाथवे का क्या काम है? TGF बीटा में क्या होता है? TGF बीटा, TGF क्या? ट्रांसफॉर्मिंग ग्रोथ फैक्टर। They are related to cell proliferation and differentiation. They may stimulate or inhibit proliferation depending on the cell type and growth factor environment. Right? WNT का क्या होता है? सिमिलर मैनर में WNT बीटा कैटेनिन पाथवे सुना होगा. It also regulates key cellular functions like proliferation, differentiation, migration, genetic stability and stem cell renewal. और नॉट सिग्नलिंग में क्या होगा? It is also cell signaling system. It also controls cell fate, proliferation and differentiation. Okay. It regulates stem cell self renewal, cell fate determination, cellular differentiation, skeletal muscle development and regeneration, binary cell fate choices. It induces the adoption of a third cell fate at the border of neighboring populations of different cells. मोर और लेस अगर कुछ भी पाथवे इफेक्ट होगा तो कहीं ना कहीं कैंसर रिलेटेड तो आता ही है सब में. बट मोस्ट कॉमन पाथवे अगर हम बात करें सिग्नलिंग पाथवे जो एपोप्टोसिस से लिंक्ड है तो वो बिना सोचे दिस पाथवे है, क्योंकि एक्सट्रिंसिक और इंट्रिंसिक पाथवे की यहाँ पर बात आती है. अदरवाइज मोर और लेस सेलुलर डिफ्रेंशिएशन और सेलुलर प्रोसेसेस में पाथवे अगर हैं ये इन्वॉल्व तो कहीं ना कहीं एपोप्टोसिस में लिंक हो ही जाता है ये. बट अगर कॉमनली एसोसिएटेड की बात हो रही है तो इट इज़ FAS ओनली. मूविंग ऑन टू नेक्स्ट. Which proteins are often referred to as executioners in the apoptotic pathway? Executioners means execute कौन करता है?
कैस्पेस.
इतना सोचना पड़ा.
साइक्लिन्स भी स्टार्टिंग में हेल्प करता है. नहीं, हाँ, साइक्लिन भी तो
एग्जीक्यूशनर पाथवे जैसे हम इनिशिएशन, एग्जीक्यूशन और क्लियरेंस की बात जब हम कर रहे थे तो यहाँ पर उन एग्जीक्यूशन की बात हो रही है. एग्जीक्यूशनर फेज में मतलब एग्जीक्यूशनर जो पाथवे होता है एपोप्टोसिस का तो उनमें एग्जीक्यूशनर्स का रोल कौन प्ले करता है? इट विल बी कैस्पेजेस. एग्जीक्यूशन फेज इनिशिएट किससे होगा?
साइक्लिन.
साइक्लिन से. कैस्पेस से नहीं होगा.
इनिशिएट तो साइक्लिन से होगा, एग्जीक्यूट कैस्पेस से होगा.
एग्जीक्यूशन फेज की इनिशिएशन की बात कर रही हूँ मैं.
अच्छा. तो
इनिशिएशन फेज की बात नहीं कर रही मैं. एग्जीक्यूशन फेज कैसे इनिशिएट होगा?
कैस्पेस से.
कैस्पेस से, राइट. So, execution phase is initiated by the cleavage of caspase 3, which results in destruction of nucleus, first of all. Then DNA fragmentation and degradation of cytoskeletal and proteins. Then chromatin condensation, formation of apoptotic bodies, expression of ligands for phagocytic cell receptors, and finally uptake by phagocytic cells. ओके. सो एक्सट्रिंसिक पाथवे होता है, इंट्रिंसिक पाथवे होता है, राइट. एग्जीक्यूशन फेज में कैस्पेज 9 का एक्टिवेशन, 3 का एक्टिवेशन, सबसे पहले 3 का एक्टिवेशन विच इज़ नोन एज एग्जीक्यूशन पाथवे विच विल लीड टू ऑल डिग्रेडेशन. एंडोन्यूक्लियस का एक्टिवेशन होगा, डिग्रेडेशन होगा क्रोमोसोमल डीएनए, प्रोटीज एक्टिवेशन, डिग्रेडेशन ऑफ न्यूक्लियर एंड साइटोस्केलेटन प्रोटीन्स विच विल लीड टू साइटोस्केलेटल रीऑर्गेनाइजेशन, एंड फाइनली formation of apoptotic bodies. So here caspases are involved. What is the role of caspase in apoptosis? ये तो रिपीट ही हो गया.
Execution of cell death program.
एग्जीक्यूशन ऑफ सेल डेथ प्रोग्राम. प्रमोशन नहीं है, एक्टिवेशन नहीं है, इनिबिशन नहीं है, इट इज़ एग्जीक्यूशन. ओके, सारे कैस्पेजेस का रोल वहीं पर ही आएगा. ऑल्दो इनिशिएट कैस्पेज 3 से हो रहा है, बट कैस्पेज 9, 3 all are present there. So they play a central role in execution of the cell death program during apoptosis and act as protease that can cleave specific proteins to trigger the morphological changes which are associated with PCD. Okay, caspases क्या है? Family of cysteine proteases that exist as inactivated precursor or pro-caspases until activated by apoptotic signals. Once they are activated, they initiate a cascade of proteolytic events which cleave key cellular proteins that dismantle the cell and lead to its demise. अब हमारे इनिशिएटर कैस्पेजेस होते हैं और एग्जीक्यूशनर कैस्पेज होते हैं. डिफरेंट कैस्पेजेस हैव डिस्टिंक्ट रोल इन एपोप्टोटिक पैथवे. इनिशिएटर कैस्पेजेस में कैस्पेज 9 आता है. Then trigger the activation of executioner caspase which is caspase 3 which directly cleave cellular substrates which are responsible for apoptotic morphology. फिर हमारा सब कुछ सेल श्रिंकेज, membrane blebbing, DNA fragmentation, so all these are driven by activation of caspases. Coming to what is the function of anti-apoptotic proteins?
Initiate cell death. Sorry. Prevent cell death. Promote cell survival.
Right. Correct. Okay. प्रो एपोप्टोटिक को बताया था ना, क्या बताया था?
प्रो एपोप्टोटिक को ब्लॉक करता है, मतलब प्रो एपोप्टोटिक एंटी एपोप्टोटिक को ब्लॉक करता है.
करेक्ट. Pro apoptotic blocks the anti-apoptotic property of anti-apoptotic proteins. Okay. तो anti-apoptotic एक्टिविटी इनकी नहीं होगी. इनका क्या काम है? एंटी एपोप्टोसिस, मतलब एपोप्टोसिस ना कराना. तो इट विल लीड टू सेल डेथ. इनिशिएट तो होगा नहीं. सो इट प्रमोट्स सेल सर्वाइवल. करेक्ट. Which of the following diseases is associated with defective apoptosis?
Alzheimer's.
Only Alzheimer?
Apoptosis, defective apoptosis leads to everything: cancer, autoimmune disorders,
Correct.
and Alzheimer's disease.
Yes.
But defective apoptosis, I don't know. Maybe.
डिफेक्टिव एपोप्टोसिस मीन्स एपोप्टोसिस नहीं हो रहा सही से, कुछ गड़बड़ हो गया, तो इट कैन लीड टू एनीथिंग. यू आर राइट. ओके, इट इज ऑल ऑफ द अबव. What happens to apoptotic bodies formed during apoptosis?
Option C.
Right, they are engulfed by phagocytes. अभी पाथवे हम जब देखेंगे तब ये पॉइंट आएगा मैकेनिज्म में. आई विल क्लियर इट एट दैट टाइम ऑल्सो. Which of the following is not a characteristic of apoptosis?
Cell inflammatory response. नेक्रोसिस में होता है.
राइट. क्या डिफरेंस है नेक्रोसिस और एपोप्टोसिस में?
एक्चुअली मतलब नेक्रोसिस लीड्स टू सेल स्वेलिंग.
राइट.
But apoptosis leads to cell shrinkage. Various apoptotic bodies are formed in the surroundings.
Right. Apoptosis does not involve any inflammatory response. नेक्रोसिस में होगा जिसमें सेल लाइसिस होगा, इन्फ्लामेशन होगा, एंड देन सेल्स डाइज एबनॉर्मली. एपोप्टोसिस तो बहुत कंट्रोल्ड प्रोसेस है जिसमें कोई इन्फ्लामेशन नहीं होगा. राइट. मॉर्फोलॉजिकल चेंजेस होंगे, सेल श्रिंकेज, क्रोमेटिन कंडेंसेशन, न्यूक्लियर फ्रेगमेंटेशन, फॉर्मेशन ऑफ एपोप्टोटिक बॉडीज. Since apoptotic cell membrane remains intact and is efficiently cleared by phagocytes, it does not trigger any inflammatory response. Which of the following is an external stimulus that can induce apoptosis?
UV radiation, stress, and growth factors.
Which option?
UV radiation, stress, growth factors.
Correct.
All the
Yes, all three. Growth factor, UV radiation, and cellular stress. Can we take a 5-minute break? I will be back in 5 minutes.
okay.
So we are here. Which of the following type of cells recognize and kill the abnormal pathogen infected cells? Which of the following type of cells recognize and kill the abnormal pathogen infected cells? It is more of immunology question.
T lymphocytes.
क्या करता है टी लिम्फोसाइट?
टी lymphocytes तो रिकॉग्नाइज करता है, लेकिन मास्ट सेल्स जो है वो पैथोजेंस को किल करता है, बट आंसर क्या होगा पता नहीं.
Answer is T lymphocytes. These are immune cells that recognize and kill the pathogen infected target cells in an orderly manner in apoptosis. So these cells accomplish the feat by binding to cell surface receptors of the infected or abnormal cell. T lymphocyte क्या होते हैं? Cytotoxic T cells. They are primarily responsible for recognizing and directly killing cells infected by pathogens. Mast cells करते हैं, they are involved in allergic reaction, not directly killing the infected cell. Okay. B lymphocyte, they produce the antibodies which mark pathogens for destruction but do not directly kill infected cells. And neutrophils, they are all primarily phagocytes, they engulf and destroy bacteria. They are also not specifically targets the infected cell. Okay, so T lymphocyte is the correct answer. When was the term apoptosis coined? Memory based question.
1972.
Yes. By whom?
Can't remember.
John Kerr, Curie, and Wiley. Kerr, Curie, and Wiley. Okay. John Kerr, Andrew Wiley, and A.R. Curie. Of the University of Aberdeen, Scotland. Molecular basis of apoptosis were revealed by studies conducted on C. elegans. So it was in 1972. Apoptosis is programmed cell death.
False.
False. Joking.
True.
It's true. It is programmed cell death. In all the questions, we have said that apoptosis is programmed cell death, in which a cascade of events leads to death of the cell. Apoptosis usually occurs in response to an external stimulus or an abnormal change in the cell's environment, on the basis of which we say that it is extrinsic and these are intrinsic. Apoptotic pathway. CD3 gene product in nematodes has homologous dash in mammals.
Caspases.
Caspases. Correct. They play crucial role in nematodes during apoptosis. The homologous family of proteins, that is caspases in mammals, are cysteine proteases, having a cysteine residue in the catalytic site. Inactivation of which of the following leads to detachment of the apoptotic cell? क्या है ये? FAK, PKB, PKC, RAF1 क्या है ये? RAF1 क्या है? Don't know. It is FAK, focal adhesion kinase. It is inactivated by cleaving action of caspases during apoptosis. Cleavage of FAK leads to detachment of apoptotic cell from its neighboring cells. PKB का inactivation. It can also inhibit apoptosis by increasing the expression of survival molecules or degrading pro-apoptotic molecules. PKC क्या करता है? Down regulation of this can lead to apoptosis. RAF1, it is an upstream kinase that counteracts apoptosis. और एक और होता है ASK1, उसका इनएक्टिवेशन होता है PKB के थ्रू, विच कैन प्रमोट सेल सर्वाइवल. ओके, तो हेयर फोकल एडिशन काइनेस, उसका इनएक्टिवेशन होगा बाय द क्लीविंग एक्शन ऑफ कैस्पेजेस, और क्लीवेज जो होगा फैक का, इट कैन लीड टू डिटैचमेंट ऑफ एपोप्टोटिक सेल फ्रॉम इट्स नेबरिंग सेल्स. Inactivation of which of the following leads to shrinkage of the nucleus? Lamin, tubulin, actin or gelsolin?
Lamin.
Yes, lamin. Inactivation of lamins by caspases leads to disassembly of nuclear lamina and shrinkage of nucleus. Lamins जो हैं वो special class of proteins हैं that make up the inner lining of the nuclear envelope. Okay. Cytokines serve as dash for apoptosis.
Substitutes, inhibitors.
Inhibitors? क्या करते हैं साइटोकाइंस? साइटोटॉक्सिक टी सेल्स को एक्टिवेट करेंगे. They serve as external stimuli. These are substances including interleukin, interferon, which are secreted by certain immune cells that can act as external stimulus. Whereas abnormalities in the DNA can act as internal stimulus for apoptosis.
एक्सटर्नल स्टिमुलस कैसे, मतलब वो तो बॉडी के अंदर रहता है. एक्सटर्नल स्टिमुलस
ये सब सिक्रीट होंगे. Serve as, it is saying serve as. जब कोई भी रिस्पॉन्स होगा तो these interleukin, interferon, these are substances which are known as cytokines, which are released or secreted, and they act as external stimulus. They are key messengers by which immune cells communicate with each other. Right?
एक्सटर्नल स्टिमुलस तो जैसे यूवी रेडिएशन, ये सब एक्सटर्नल स्टिमुलस हुआ, पर ये तो बॉडी के अंदर ही सिक्रीट होता है, तो एक्सटर्नल स्टिमुलस मीन्स there must be internal stimulus. Because they are secreted within the body.
Okay, I will check it again then.
But cytokines also help in inflammation. So I
Yes,
said that
yes.
it can be serves as inhibitors for apoptosis, because apoptosis leads to cell shrinkage.
So it might be best response how they could be best served as for apoptosis. So in that case, it might be external stimuli, but I will.
External stimuli might be in one case that when from outside the pathogens are coming, then cytokines are secreted, means and this leads to the death of cell or
Yes.
something.
Yes.
Then only they can be act as external stimulus, because by just present inside the body they can't be external stimulus. uh, Outside the pathogens are coming, and then the interferons and or the interleukins are getting activated by the cytokines, and then they
So
destroy the pathogens.
they are secreted, but they will serve as external stimuli.
Okay.
Okay, it is saying that they are secreted by immune cells, although they are secreted internally, but they serve as external stimuli. It is whole pathway, right? So, like, they are also involved in regulation of inflammation, as you said, in immune response, tissue repair, right? So basically they act as messengers, we can say. They trigger specific signals either in the same cell, endocrine, or distant cells. So in that way it might be like external stimuli they are saying. As we have cytokine receptors as well, which are extracellular. Right? I will check this question again. Although this serves as external stimuli, best possible answer is this. Okay. Which of the following is an extracellular messenger of apoptosis?
Extracellular means outside the cell, but TNF are present on the cell membrane. TNF are present on the cell membrane.
Extracellular messenger of apoptosis. When we talk about intracellular stimuli, it will also cause apoptosis. But intracellular stimuli, I mean, it will be nutrient deprivation or any genetic change or any kind of stress like osmotic or hypoxia. But when we talk about cytokines, these are extracellular. And here, extracellular messenger of apoptosis, it is TNF. We have TNF related apoptosis inducing ligand also, which is trail और FAS जो हमने बात किया था, FAS cell surface death receptor. Okay. So these death receptors are transmembrane proteins with cysteine rich extracellular domains. तो यहाँ पर एक्स्ट्रासेलुलर मैसेंजर जो है वो TNF है, ट्यूमर नेक्रोसिस वेक्टर. It has the ability to ablate tumor cells and is secreted by the cells of immune system in response to adverse conditions. Which of the following is also termed as a death receptor? डेथ रिसेप्टर कौन सा है? TNF की तो हमने बात कर ली, ट्यूमर नेक्रोसिस फैक्टर. अब कौन सा वाला है? 1 2 3 4. TNFR1. Death receptors are so called because of the presence of a death domain which is responsible for killing an infected cell. TNFR1 is one such example. It plays an important role in induced apoptosis. Okay. साइटोप्लाज्मिक पार्ट में इसका एक डेथ डोमेन होता है जो रिस्पॉन्सिबल होता है प्रोटीन प्रोटीन इंटरेक्शन के लिए एंड इट अलाउज death receptors to interact with other proteins that also have death domain, and these are expressed on most cell types. TNF signaling में होगा and it can induce either apoptosis or proliferation. TNFR1 is a trimeric receptor. Is it true or false? Tumor necrosis factor receptor 1. It is a cell surface receptor and it forms trimeric structure. So it is true. It is a trimeric receptor. It contains a domain of 70 amino acids to which the tumor necrosis factor binds and brings about a change in the shape of the receptor. After there is a conformational change, proteins are recruited. The last proteins to involve in the apoptotic pathway induced by TNF are TNF के apoptotic pathway में last proteins कौन से involved होते हैं? Remember? It is procaspase 8. The last proteins that get involved in the apoptotic pathway triggered by the tumor necrosis factor, they are procaspase 8, which are precursor of caspase proteins. So these procaspases contain an extra portion that must be removed by catalytic action. I hope you know what is difference between procaspase and caspase. Which family of proteins regulate the intrinsic pathway of apoptosis? Which family of proteins regulates the intrinsic pathway of apoptosis?
Caspase 2. Yes, ma'am.
What? What is the answer?
Caspase 2.
Caspase 2. Intrinsic pathway.
BCL2.
It is BCL2, right? BCL2 family जो होते हैं उसमें regulator proteins होते हैं. These proteins are present inside the cell, that is they are intrinsic in nature. They carry out apoptosis in damaged or old cells. Okay, so these regulate the intrinsic pathway of apoptosis. The key control point in apoptosis, which is a molecular program that destroys targeted cells. BCL2 में हमारे 4 कंजर्व्ड BCl2 होमोलॉजी डोमेन्स होते हैं, BH1 2 3 4, जिसमें 3 जो होता है विच इज़ असेंशियल फॉर रेगुलेटिंग सेल डेथ। ओके, BCl2 फैमिली प्रोटीन्स में दो अलग तरीके के फंक्शंस होते हैं, एंटी एपोप्टोटिक जो हमने पढ़े थे और प्रो एपोप्टोटिक। एंटी एपोप्टोटिक में क्या था, विच प्रोटेक्ट सेल्स अगेंस्ट एपोप्टोटिक स्टिमुलाई, जिसमें BCl2 था, BClXL था, MCl1 भी था। और प्रो वाले में दे एक्टिवली किल द सेल बाय इंड्यूसिंग MOMP, माइटोकॉन्ड्रियल आउटर मेम्ब्रेन परमिएज। एग्जाम्पल क्या थे, बैक्स, बैक, बिड। याद आया? याद है? आज ही पढ़ा। ओके, विच ऑफ द फॉलोइंग इज इन्वॉल्व इन द इंट्रिंसिक पाथवे ऑफ एपोप्टोसिस, विच ऑफ द साइटोक्रोम?
Cytochrome C.
It is cytochrome C. It is the compound which is involved in the intrinsic pathway. It is also component of ATC and mitochondria. As the process of apoptosis initiates, the cells lose contact with the environment. Apoptotic bodies can be recognized by the presence of dash on the surface. ये तो पता होगा। किसकी बात कर रहे थे हम? याद करो। It is phosphatidylserine. Apoptotic bodies can be recognized by the presence of phosphatidylserine on their surface. It is a compound which is usually present on the inner surface of the plasma membrane. So, it is involved in the recognition and removal of apoptotic cell by phagocytes. Okay, so these questions have been covered today, and I can show you the pathway which I was talking about. Screen is not being shared.
No. Or you can keep the link in the chat box and then I will see it.
Yes, it's shared. No, not shared. Again, there's the issue.
Which type of questions can come in CSIR net?
What?
This type of questions can come in CSIR net.
CSR net
Mm.
related to this cell bio.
Mm. So,
Caspase is related. Caspase is apoptotic pathways, extrinsic, intrinsic. They are
and
factors.
is NPTEL courses means from the NPTEL course can we appear for CSIR net?
Yes, it is helpful. Not questions exactly can come,
No,
but
but the concepts means
concepts is clear. Like I want to share the PPT of apoptosis, necrosis, and other factors, caspases, and extrinsic and intrinsic pathway. But it is not being shared. I will then show in next class, and also next will be the last class, and also I have applied for that extra class as well. So if next class we won't be able to complete it, in the last class we can cover the concepts of all the classes till now, right? So it will be helpful for your CSR net.
same day.
Not same day. I think I have applied for it on some other day. I will let you know in the next class. So it will be on 14th March. It will be week 8. I think I have applied for 15th March, that is Saturday. Maybe on the next day, because we have to complete the course. I will let you know on the 14th only. Okay. And also week 1 and 2, there's some issue in the recording, so I might have to take those classes again. So I will let you know. You can share your email ID. I will let you know whenever I will be taking or you are available. Right, you can share your email ID in the chat box. I will let you know whenever I will take that extra class, and also the two classes for which my video recording was not completed. I might take that extra class as well. So I will complete the concepts in that, and it will be useful for your CSIR net. Okay, so please share your email ID. Are you sharing?
बाकी के पेमेंट्स आपके पास है?
टुप्पो क्या किया था? सेमी क्यों नहीं?
हमारे पास क्योंकि नहीं है 2OG और
हो सकता है।
CK9 एक साथ ही हम रखे थे आपका
हो
सारा,
सकता है।
तो
हो गई बस एक
अच्छा,
बार
हो
ये,
गया?
हाँ बस
ठीक।
एक बार ईमेल आईडी।
हम तब से टेबल साफ सफाई कर रहे
हाँ
हैं।
ठीक है। Okay, I am ending the class then. If you're comfortable, you can share your email address. I will let you know when will the extra class will happen. Okay. Good night. Thanks for attending.